First, it's my great honor to present this global research of 2022. My name is Yuanping. I'm from FAFU. I'm a PhD student. Today, my talk is about the identification of metallophosphate that confers resistance to arsenic. So metallophosphate is a gene. And as we all know, the arsenic is a one world problem. Even all over the world. I don't know how to do this. And we can say from this picture, we can say that arsenic contamination in China, especially in all in the world, is very heavy. Very serious. It is widely distributed in the world. And at the same time, with the research getting more deeper, we can see lots of microbials can drive the arsenic from the organic one to the inorganic one and turn back to the inorganic one. And through the methylation or the arsenic oxidation or the reduction or the demethylation, the arsenic has different shapes, different species. And most of this, the arsenic exists in the world, in nature for the arsenite and arsenate. And in the microbial community, and we can say there are lots of the enzymes who who drive the arsenic different shapes, transformation, and we can see from the arsenite get inside from the cell, get inside to the cell from the environment by the group P and also the aquaporin transporter, and then once it gets inside, it can be reduced. It can be oxidized to the arsenate, which is the less toxicity to the microbials. And at the same time, the arsenate can be reduced to arsenate. And the arsenic inside the cell can be methylated by SM to the DM3 and AM3 and TM3, and while the TM3 is volatile, it can reduce the arsenic concentration in the bacterial cells. And at the same time, the arsenic can be oxidized to the arsenate. And it can be methylated or transferred to the organic arsenate. The organic arsenate can be oxidized into the organic arsenate. While the arsenate is less toxic to the microbials. And during the research, we found the organic arsenic is very important, and it has been widely used as the herbicide and also the food addictive, which helps the expression of the dispersion of the arsenic in the microbials. However, the microbials also can do the biotransformation of the organic arsenic. And for us, because we are very interested to know how the microbials deal with the arsenic in the heavy metal contaminated environment. So we got the strain from the heavy metal contaminated mining, and we got and then we do the genome sequence and also the RNA sequence to get a cluster of the different expressed genes, and then we got lots of targeted genes. The targeted genes, then we use the heterogeneous expression to verify the function of some putative arsenic genes. And this is what the result, what we got. From the mining, we got one strain named Mucilaginibacter Rubeus P2. And when we do the metal minimal inhibitory concentration of our Mucilaginibacter P2 strain to different metal or materials, we can easily see the strain P2 shows very high resistance to the copper and also the arsenic and also the zinc and also even much higher resistance to the gold compared to the cuprobidos and the CH34. Yeah, this is a very well studied strain for the heavy metal resistance. Yeah, and during our RNA-seq analysis, we found there are lots of the genes who is upregulated by the arsenic stress of the 0.2 millimolar of arsenite added, and we can see, yeah, the SR, lots of the 4SR and the 3SU and SN and ACR and SC and ACR3 and metallophosphate 1, metallophosphate 2, yeah, and after we analyze more into detail from the complete genome, we found, yeah, there are 4 S oblongs and 3 of them we can easily see is very similar to each other. So then we have our hypothesis, yeah, how this strain triplicated the S oblong in this hash experiment, in this hash environment. So then we got well, because, yeah, until now there's no study focused on the metallophosphate on the arsenic resistance. So we did the blast search for our metallophosphate 1 and metallophosphate 2. These 2 genes, we can easily find those 2 genes always sit together. We always sit together with ACR3 in the strain Mucilaginibacter and also as Actinobacter and Flavobacteria. All the strains located in the final of Bacteroides, and we all know the Bacteroides final is a very abundant bacteria in human gut. Yeah, and then we are very interested to see, yeah, maybe this metallophosphate 1 and metallophosphate 2 can do something to the arsenic. Yeah, so then we got our metallophosphate 1 and metallophosphate 2 separately, clone them into the strain Ecli AW3110, which is an arsenic oblong deleted strain, is very hypothetical to arsenic. And then when we put the metallophosphate 1 or metallophosphate 2 inside the AW3110, we can see, yeah, under the stress of different concentration of arsenite, yeah, we can see, yeah, with the metallophosphate 1 get inside, yeah, the strain grows much better than the empty plasmid in AW3110. Yeah, this tells us, yeah, the metallophosphate expression in the AW3110 confers resistance to the arsenite and also the loxosome 3 and also the metallophosphate 3, also the arsenate. Yeah, and the loxosome 3 and the metallophosphate 3 is the organic arsenite, and the arsenate is the inorganic arsenite. Yeah, and then all this tells us, yeah, the metallophosphate 1 gives resistance to the organic and inorganic arsenite, and the same results as the metallophosphate 2, which I didn't put inside. Yeah. And then we get our conclusion is that, yeah, after our verification of the genes named metallophosphate 1 and metallophosphate 2, and we found it gives resistance to both organic and inorganic arsenic in the strain Mucilaginibacter Rubeus P2. And because the organic arsenic is widely distributed in the microbials, and it can enhance the competition of microbials. Yeah, and it has been treated as an antibiotics. Yeah, so we get a conclusion that the metallophosphate, Mucilaginibacter Rubeus P2 confers a high level of resistance to both organic and arsenic, and it can be often used in the interbacterial warfare. Yeah, and so thank you for your listening, and that's all my presentation. I hope you can see the point and get a little bit my message.